All right, so hi, thanks for coming. This was a student project, and students do what students do. They graduate, and then they give me a spreadsheet with all their data, and they all got jobs, and so I get left to present the project. This was a project conducted at Mill Creek, and I hope you caught Lathrop's talk on Tuesday about variable density thinning at Mill Creek, but this was in those same stands. We studied bear damage. We did two things. This was a two-part study where up at Mill Creek we tallied bear damage in 3 different silvicultural prescriptions, and so the prescriptions that we studied were a heavy thinning, and this thin was down to maybe as much as 110 trees to the acre. It was a heavy thinning, and Lathrop described these prescriptions on Tuesday. A high density, I'm calling it a lighter thin, but it's still a lot of trees were cut in the high density thinning, and then finally a no thin control. And so we counted bear damage in these 3 treatments. The second part was we collected increment cores on bear damaged trees, and there we were interested in how fast were the trees growing at the time of damage and when did the damage occur, and also we had a peek at phloem thickness. So another look at the map. The coloured polygons are different silvicultural treatments that have been applied to these crowded regenerating stands, and we focused on 2 treatments, but there's opportunities to study bear damage in a whole lot of different silvicultural prescriptions. We just focused on treatments that were of interest in industrial forestry as well as restoration, these pretty common PCT treatments. The study, we randomly selected stands to study, and we got a really broad geographic coverage in this watershed. So we're talking about stands that were up to 16 miles apart. This was our transect sampling scheme, and so starting from the road on the left, much like a search party would move through an area. The students formed a belt transect and just tallied trees as they moved through the forest, and they were counting incidences of damage on the trees. They were recording species and also size. So there's a sort of by size class tallies of bear damage or not bear damaged in conifers. And they would move through the stands, section 1, section 2. These sections are a chain long, if that helps. And so that gave us an idea not only of bear damage, the rate or the percentage, but also whether that rate was changing the further we got from the road. And that was sort of the novel contribution of this study, was looking at ease of access, or how close to the road, whether the damage was changing as you get further away from the road, where we know the bears like to travel down the roads. And then once they made it through, it wasn't an easy walk in the park, because after these PCT treatments that were conducted pretty late, there was a lot of slash. So it was more of a scramble and crawl to do this sampling. I'm told I wasn't there. I get this job. Here's the data, just size class along the bottom, and then proportion of trees damaged. So just glancing at the left-hand side of the graph, you'd see that in the smaller size classes, these are even age stands, all about the same age. So these are the smaller trees in those even age stands are only being damaged up to about 10% of that size class is being hit. Very few small trees being hit. And then out of those bars, let's see if I can get a mouse going here. Nope, no mouse. On the left is Douglas fir. There's sort of a group of 6 bars. On the left would be the Douglas fir and the different treatments, and on the right would be the redwoods. So look to the right then at the largest sizes. That's 40 centimetres and above. 40 centimetres would be a 16-inch DBH class and above. On the right-hand side, you've got 6 bars. The first 3 bars are Douglas fir. So Douglas fir in the black is the control, so no thinning, very low rate of damage. And then as we increase in the thinning intensity, we're seeing a much higher proportion of the Douglas firs damaged in that heavy thinning. Reading across, that's over 50% of the Douglas fir was damaged. And then in the redwoods, a slightly different pattern there, that redwoods were being damaged no matter what, especially the large redwoods. But there was a thinning effect there. So what we had to do was we had to sort of tease out some other factors. We had to do an analysis of, we had to model the probability of damage. And so this is a probability model. And so on the left-hand side, you would read across and you'd say, okay, a tree of 30 centimeters DBH, that's your 12-inch DBH size. If it's a redwood, you can expect 60%, 0.6 proportion damage. So you're reading up the tree size and across. So this kind of has management application. You can kind of figure out, okay, this is the kind of rate I can expect in damage to redwoods if I do this low density thinning. Douglas fir, much less likely to be damaged. So this model, we can also use the model to look at what was the effect of the thinning treatment. So this graph is for redwood only now, and what we're seeing is basically the thinning, the low density thin is doubling essentially the probability or the proportion of trees being damaged, redwoods. And the high density thinning has an intermediate effect. And again, with DBH on the bottom, the damage is being focused in the larger size classes. Lastly, distance from road. So now we're going to hold constant. We're just going to look at the high density thin. High density thin is probably the most commonly applied treatment, thinning down to 16 by 16 feet. And what we're seeing here is the damage to redwood. You see the strong effective size class, right? DBH. The larger trees are much more likely to be damaged. That's on the right-hand side of the graph. But as we go further from the road, our damage rate or our probability of damage drops. And it doesn't drop linearly, it drops really fast. And so what we're seeing is the bear is concentrating the damage within the first chain on either side of the road, roughly speaking. And then after that, the damage tails off and kind of stabilizes to a steadier level inside the stand. Move on now to the second part of the study, which was the increment core analysis. And so here, this is we took some cookies just to kind of show what it looks like inside a bear damaged tree, but we collected increment cores and we cored in from both sides. So we cored through the damaged side and the live part on the back. And the growth continues on the live side, but it ceases on the dead side, obviously, and so that is convenient for us because we could count the rings and say, okay, in this instance, there's 10 rings on the damaged side and there's 11 growth rings on the live side, so we deduce that the damage occurred 1 year ago. And that's pretty handy to know when the damage occurred because then we can say, oh, it was damaged after the thinning treatment, not before. So we're detectives now. This is washed out, but I believe you have a nice picture of it on the left-hand side, I hope, is an increment core showing, we ran this through Windendro. Windendro you scan, and then the software is smart, and it figures out how wide the rings are, the growth rings, and the phloem. And we were quite interested in how fast was the tree growing and whether the thickness of the phloem, which is what the bear is feeding on, how thick that layer was in relation to growth rates. That's something we were curious about. Because in the last few slides I've shown you how tree size and damage were related, but really tree size was kind of a surrogate for growth rate because this is an even age stand and the bigger trees were the faster growing trees. So we looked at the last year's growth in relation to phloem thickness and the last 5 years' growth as a sort of an averaging of how fast this tree was growing lately. Here's the modelling results of that. So diameter, tree size, dBH on the bottom, and phloem thickness on the y-axis. So as you get in the larger trees, they also have thicker phloem. This is a radial measurement. So not only does a large tree have more phloem around it, but it's thicker in general. And then as we go up the lines, those are growth rate lines. And so the lowest line would be a very slow-growing tree. And as you go up the lines, it's telling you that a tree that's one of the more faster-grown ones that we sampled had much thicker phloem. And that phloem, lastly, this is a sort of a finer point, but the slow-grown trees seemed more sensitive to tree size than the fast-growing trees. Fast-growing trees had thick phloem, no matter what. That line's kind of flatter across the top. Yeah, so that's the outcome of a regression analysis with 3 variables. And lastly, looking at growth of radial increment, how fast were these trees growing at the time of damage? We've got bars. These are growth rates, so radial increment. So faster growth, taller bar. And if it's a damaged tree or an undamaged tree, this is how fast they were growing. Now these are paired samples. So what we did was we went and found a damaged tree and then looked within a very narrow search radius around it for an undamaged tree of similar size. We're interested in what might be different between the tree that the bear chose and the tree that the bear did not choose to damage. So these were sampled within 3 and a bit meters, 12 feet or so, so very close, tightly paired, same growing conditions, well, as best as we could do. So we're seeing here that in the controls, the unthinned controls, the damaged trees were growing much faster than the undamaged trees. So growth rate was it really looked like really strongly tied to probability of damage. In the high density thin, those are the middle 2 bars. High density thinning, we're seeing that the damaged trees also were growing faster than the undamaged the paired trees. And the same was true in the low density thin. So the bear had a choice, and in most cases it chose the trees that were faster growing. That's what this data is showing, but also that there's a bit of noise there. Those are those error bars. So to wrap up, Redwood has a higher probability of damage than Douglas fir. Check. You can use these graphs, which the graphs will be in a, I'll give you a link to a publication that we have, and also check out the Symposium proceedings where we put the phloem stuff in as well. But you can go to these graphs and read across and kind of get an idea of, okay, how much damage can I expect? And maybe you want to adjust your prescription. So maybe you want to keep a few extra trees when you're thinning in anticipation of this damage that we're trying to help you predict. The increment cause and our sort of fancy dating process confirm that 100% of the damage in our samples occurred after the thinning treatments. Damage was exclusively after thinning, and this is even in the unthinned stands. If that makes sense. Think on that. Bear damage definitely correlated with rapid diameter growth, and I can't make a link between the last 2 points, but we observed that the larger redwoods, the faster growing redwoods, had thicker phloem. Now, I can't make a causal link there, but I'm just pointing out that these are anatomical observations that we made. And so really, if I could do this again, and it would be good to, I think we should think about looking at the relationship across a wider range of tree sizes. I think we should look at even-aged versus uneven-aged management, because there's this idea that the tree regenerating in partial shade versus full sun might have different taper, different phloem thickness, and it may be more or less attractive to bears. And that's what I mean by a variety of stand structures. So even-aged, multi-aged, or in different silvicultural systems. So if anybody wants to partner up on this, yeah, and the next talk is with Ken. We're excited about this. Ken Fulgham's going to give us an overview of bear damage. So I think we have time for, do we have time for questions?
We have lots of time for questions.
Cool.
Any questions? Yes, Robert.
So I'm just curious about the phloem thickness. So presumably these trees that are growing faster have more sugar, so the bears they can smell that? Are they sensing it?
Well, who would know that here? Somebody must be more qualified than I to talk about
Sure.
bears. Tim. I'm looking at Tim.
So in other words, then, it's not necessarily so much a learning process of sows teaching young?
Well, the Humboldt research that came out several years ago through DNA analysis found that females were the first to enter an undamaged stand, and the females, and again, you have to remember when this is happening, these animals are starving, and so it makes sense that a lactating female is desperate to find nutrients for her young. So they found rather conclusively that females are the first to move in. So I don't know if it's learned behavior or just luck behavior, but the baby's watching mom. And then the other thing too, we've also seen how bears will taste a tree, right? They'll strip off a piece, come back 2 weeks later, and then hammer the tree a second time. So there's different cues that they're getting by sampling.
I actually have a video of a female up the tree with the two cubs, showing them how to strip it with a big liquid.
Any more questions? Yeah, one more.
Your curve is moving upward. It hasn't peaked at 50 centimeters DVH, and it's been my experience that the larger redwoods are less likely to take on bear damage. Do you know at what point that curve starts to decline again in terms of DVH?
Sure. So the question is, is there a decline in bear damage with increasing tree size beyond what we sampled? And so we sampled in even age stands, and so these trees here, I didn't go past 50 centimeters with this graph because we didn't sample any trees, or many trees above that size. So no, we don't know what's happening with the larger size trees. And I was kind of driving at that with my last point, was we need to study a wider range of stand ages and stand structures. But does anybody here, have they seen damage in larger trees, or would they want to add to that?
They just climb further up the tree.
So we have a comment here, they just climb further up the tree if it's a larger tree.
That's what we're seeing in Mendocino County.
Gordy?
And Linwood knows that. I mean, you start with these big trees, but they get up within that 16 to 20 inch DVH range up there.
The area that I'm thinking of, I would say 75 to 80% of bear damage was in trees larger than 3 feet in diameter. And they climbed up until they got to about 20 inches, and from there, stripped it up.
Yeah. Okay, let's keep moving. Pascal, thank you so much for stepping in.